Hello students, today you are going to study chapter number 7, that is cell division. Chapter number 7, cell division. So let us see first a definition of cell. So cell can be defined as it is a basic structural functional unit of life. I repeat my sentence. Cell can be defined as it is a basic structural functional unit of life. In this chapter, cell division, you have to study 3 points one by one. First one is cell cycle, second one is mitotic cell division or mitosis, and the second one is the meiotic cell division. Let us see what is cell cycle. Okay, so sequential changes occur in the life cycle or in the life of cell is called as cell cycle. I repeat my sentence. Cell cycle can be defined as sequential changes occur in the life of dividing cell is called as cell cycle. Now that cell cycle is divided into two parts. First one is interphase and the second one is actually mitotic cell division or meiotic cell division. So let us see what is cell cycle. Part of the cell cycle is interphase and the second part of the cell cycle is a mitotic cell division or a meiotic cell division. Let us see first part of this cell cycle, that is the interphase. Now this interphase is further divided into three sub stages. First one is G1 S phase and the G2 phase. Now just see the first one is G1 phase. Okay, so this phase is also called as a gap phase because this is immediately after a cell division. It means that after cell division, cell enters into a G1. First G1, G0 is also there, but ultimately cell enters into the G1, that is called as a growth phase 1. This shows in this phase cell shows a proper growth, that is called as a cell growth. The next one is this shows the normal function, cell function, in the duplication of organs present in the cell or the cell organelle duplication or the duplication of cell organelles. So this is one. This is first one and this takes probably the 8 hours. This shows also RNA synthesis. Okay, that's why this G1 phase is called as a first growth phase of the cell cycle. Now just move towards the next and the most important part, that is the second phase of the interphase, that is the S phase. This takes probably 6 to 8 hours. Okay. In S phase, this S phase is also called as a synthesis phase because DNA duplication takes place only, duplication or replication takes place only in S phase. That's why this S phase is called as a DNA replication phase. Simultaneously, histones are required for the duplication of DNA. So histones are also synthesized in this particular phase, that is S phase. So this is most important phase of this cell cycle. So second part or the second subphase of the interphase is called as a S phase, which is also called as a synthesis phase. Now just move towards the next one, that is called as a G2 phase or second growth phase, that is G2 phase. So you can see this G2 phase, this takes probably 2 to 5 hours. This is a most important, again all these are important phases, but this is also third and the last phase of this one, that is interphase. So this is called as a second growth phase. This takes probably 2 to 5 hours. In this phase, cell shows higher metabolic activities, as well as in this phase cell synthesizes a RNA. Okay, so this in this phase of the interphase shows high metabolic activity as well as RNA synthesis, or simply you can say cell undergoes many metabolic activities as well as cell synthesizes RNA. Okay, so this is third phase, subphase of the interphase. As I said earlier, this entire cell cycle shows two main phases. First one is interphase and the last one is actually mitotic cell division or the meiotic cell division. Understood? Now just move towards the next one. After this G2 phase, cell enters into the actual cell division phase. That is mitotic cell division or meiotic cell division. In this, cell divides in mitotic cell division, cell divides into two daughter cells. Let us see the phases, subphases of the mitosis. First one is prophase, metaphase, second one is metaphase, third one is anaphase, and the fourth one is telophase. Again, this mitotic cell, let us see what is mitotic cell division. Mitotic cell division takes place only in somatic cells or body cells or a vegetative cells, in plant cells, okay, or in plants. Okay, so this mitotic cell division, this is also called as, mitotic cell division is also called as a duplicative cell division, because in mitotic cell division, cell duplicate once and divides once. It means that whatever genetic material is present, duplicate once and it divides once. This mitotic cell division, again it is divided into two sub stages. First one is karyokinesis and the second one is cytokinesis. So karyokinesis, that word itself indicates its meaning, that is the karyokinesis. Karyokinesis means a division of chromosome. Karyon. Karyon means nucleus, and the kinesis means division. Again, that karyokinesis includes four different stages, that is prophase, metaphase, anaphase, and the telophase, followed by cytokinesis. This word itself indicates its meaning, that is the cyto. Cyto means cytoplasm and kinesis means division. So this mitosis involves two stages, actual division of nucleus, and the second part is division of is cytoplasm. Okay, let us move towards the actual mitotic cell division. Now you can see this is an animal cell, because there are two types of division found in plants and the animals. As far as animal cells are concerned, you can see astral type of mitotic cell division. Astral mitotic cell division. It means that fork-like structure develops in the cell during the cell division. That's why that type of cell division is called as a astral type of mitotic cell division. So you can see the aster. This is aster. It looks like a fork-like structure, and this is called as an aster. Are you getting my point? Okay. So this is called as a astral type of mitotic cell division, which is mainly found in animals. While this type of aster is not developed in the plant cells. That's why that type of cell division in plants is called as a anastral type of cell division. Understood? So this is simply regarding the astral type of mitotic cell division and the anastral type of mitotic cell division. Let us see the mitotic cell division or mitosis in animal cell. Okay, when cell completes its interphase, it enters into the mitotic cell division or meiotic cell division. So today you are going to study first mitosis, then we will study the meiotic cell division. Let us see the first phase, that is the prophase. Again, this one is divided into early prophase and the late prophase. Early prophase and late prophase. Let us see regarding the prophase. In this prophase, nucleolus, you can see this is a cell membrane, that is called as a cell plasma membrane. Then you can see a cytoplasm, and this is a nucleus showing the chromosomes. Okay. So let us see what is prophase. In this prophase, nucleolus, this is called as a nucleolus, present in the dark stained body present in the nucleus is called as a nucleolus. So nucleolus as well as a nuclear envelope start disappearing. So first indication of this prophase is this nuclear envelope. This is called as a nuclear envelope or the nuclear membrane, as well as this nucleolus start disappearing. So you can show like this dotted nuclear envelope. Okay. Then this chromatin network undergoes a condensation. This reveals into the definite number of chromosomes. So now this nucleus shows definite number of chromosomes due to the condensation in chromatin network. In this, after condensation, each chromosome appears just like a bivalve structure, okay, which is connected to the centromere. So you can see the centromere where the two chromatids are connected with each other. Okay, so this is simply regarding the first phase, that is the prophase. Okay, now just move towards the next one, that is the metaphase. So this word itself indicates its meaning, that is metaphase. Okay, so meta means middle, and phase means stage. Now you can see in this diagram, in this cell, this is a fork-like structure which is called as an aster, and these two asters are connected with each other by these spindle fibers, and these spindle fibers are proteinaceous contractile fibers. So all these proteins fiber undergo contraction, okay, and due to this contraction, these divided chromatids pull towards the opposite pole. So these chromatid fibers are proteinaceous, okay, and these two fork-like structures are known as asters, and these two asters are connected with each other by these contractious or the proteinaceous contractile fibers. Okay, what is actually metaphase? So this word, as I said earlier, this itself indicates its meaning, that is the meta and the phase. Meta means middle and the phase means stage. In this metaphase, all these chromosomes get organized. All these chromosomes get organized in the center or equatorial plane. That's why this phase is called as metaphase. So all these chromosomes get organized on spindle fibers at center or middle or equatorial plane. That's why this is called as a metaphase. Let us move towards the next, that is third phase, that is called as a anaphase. Now in this phase, you can see in this anaphase, a centromere, as I said earlier, a single chromosome or a complete chromosome consists of two chromatids, and these two chromatids are held together by a middle point, that is called as a centromere. Now in this phase, centromere of each chromosome divides into two chromatids, and these divided chromatids are pulled towards the opposite pole on the spindle fiber, because these are contractile proteinaceous spindle fibers which are connected to the opposite poles or the asters. Okay, because this is animal cell, no? Am I right? Okay, so this is simply regarding the anaphase. In this anaphase, सेंट्रोमियर ऑफ ईच क्रोमोझोम, इथे बघा, इथे हे सेंटरला अटॅच्ड आहेत, इथे सेंटर पोझिशनला ते आलेले आहेत, आणि या सेंटर पोझिशनला त्यांनी काय केलेलं आहे, बरोबर सेंटरला म्हणजे इक्वेटोरियल प्लेनला आलेले आहेत. पुढच्या याच्यामध्ये आपल्याला दिसतात की ते अशा पद्धतीने ओढले गेलेले आहेत, म्हणजेच एक क्रोमोझोम आहे त्याचे दोन क्रोमॅटिड्स डिवाइड झालेले आहेत. म्हणजेच काय, या सेंट्रोमियर या याच्या इक्वेटोरियल पोझिशन वरती असताना हा क्रोमॅटिड आणि हा क्रोमॅटिड हे एकत्र होते, म्हणजेच ह्या दोघांचा एक क्रोमोझोम झालेला होता. समजतंय का? तर अशा पद्धतीने याच्यामध्ये या एनाफेज मध्ये, इन दिस एनाफेज, ए सेंट्रोमियर ऑफ ईच क्रोमोझोम डिवाइड्स इनटू टू क्रोमॅटिड्स. नाउ these divided chromatids pull together towards opposite poles on these contractile proteinaceous spindle fibers, okay, which are connected to the opposite poles. Okay, this is called as a anaphase. Now just move towards the telophase. Telophase. In this telophase, two cells are formed and a nuclear envelope and a nucleolus. Both these things reappear at the end of telophase. Actually, this telophase is further divided into two parts. That is telophase, early telophase and the late telophase. Understood? So at late telophase you can see nuclear envelope as well as nucleolus. Both these things reappear at the end of late telophase. Understood? So this is a fourth stage of the karyokinesis. This is followed by cytokinesis, in which in animal cell a furrow-like structure is developed at the center, and this increases towards the center, and this results into the formation of deep furrow, and because of this two cells are formed. Now these cells are known as daughter cells. In case of plants, a cell plate is formed at the center, or cell plate formation takes place at the center at the end of cytokinesis in the plant cell. A basic difference between the plant cell division and the animal cell division is a fork-like structure only develops in the animal cells. A fork-like structure. You can see a bicycle and a fork, a center, and the center is connected with the forks. Understood? So this type of structure develop to the opposite poles of the animal cell, while you cannot find this type of structure in the plant cell. Because of development of such structure in animal cells, this type of cell division, that is mitotic cell division, is called as astral mitotic cell division. As far as plant cells are concerned, you cannot find this structure, that is fork-like structure, that is astral. So that's why that type of cell division is called as anastral type of mitotic cell division. ॲनिमल मध्ये काय होतं, ॲनिमल सेलच्या सायटोकायनेसिस मध्ये असा हा फरो हा इकडे डीपली इन्क्रीज होत जातो, आणि एक वेळ अशी येते की या याच्यापासून या फरो डीप आतमध्ये सेंटर पर्यंत गेल्यामुळे एकापासून दोन सेल्स तयार होतात. तर प्लांट सेल्स मध्ये काय होतं, इन प्लांट सेल्स ए सेल प्लेट फॉर्मेशन टेक्स प्लेस ऍट दी सेंटर. सेल प्लेट तयार होते आणि ती मग इकडे एक्सटेंड होते अशा पद्धतीने, आणि मग एकापासून दोन सेल्स तयार होतात. दॅट इज कॉल्ड एज ए cytokinesis. So mitosis is divided into two parts: a karyokinesis, that is a division of nucleus, and the second part is a division of cytoplasm. किती वेळ झाला? किती वेळ झाला? अगं किती वेळ झाला? ए किती वेळ झाला? किती आहे? किती मिनिटं आहेत?
सांगते एवढ्यात कसं सांगते.
Okay. So today you have studied, yes, that is after cytokinesis you can see two daughter cells.
16 मिनिट आहे.
Yes,
16 मिनिट.
two daughter cells after a cell division. Okay. Then there is one more point, that is a amitotic cell division. Okay, amitotic cell division takes place in the abnormal cells. Okay. Or the unicellular organisms in which cell divides unequally or maybe divide equally into the two daughter cells. So that sort of cell division is called as a amitotic cell division, while this one is normal cell division in which cell divides into two daughter equal cells. Okay, so I repeat once again this entire life cycle, cell cycle, and then we will see a overlook on this part, that is the mitosis. As I said earlier, this is a life cycle of the cell. It is also called as a cell cycle of the cell. Understood? Now this cell cycle is divided, it is broadly divided into two parts. Okay, first part is called as interphase and the second part is mitotic cell division or meiotic cell division. Now this interphase is, let us see what is cell cycle. सेल सायकल म्हणजे त्या सेल डिव्हिजन, सेलमध्ये डिव्हिजन होण्या अगोदर त्याच्यामध्ये जे सिक्वेन्शनल किंवा त्याच्यामध्ये काय जे बदल होतात, त्याच्यामध्ये काही ज्या घडामोडी होतात याला सेल सायकल असं म्हणतात. सो सेल सायकल कॅन बी डिफाइंड ॲज सिक्वेन्शनल events. Sequential events take place in the life of cell. In the life of cell is called as cell cycle. I repeat my sentence. Sequential or whatever changes take place in the life of cell is called as a cell cycle. Now this cell cycle is broadly classified into interphase and mitotic cell division or meiotic cell division. Now this interphase is further divided into three sub-phases: G phase, S phase and the G2 phase. This G1, this part of this cycle, cell cycle is called as a G1 phase. G1 phase, this is also called as a first growth phase. Okay. In this phase, cell shows a growth as well as normal cell functions, and this shows duplication of cell organelles and simultaneously synthesis of RNA. Understood? Then cell enters into the second, that is the S phase. So this S phase is called as a synthesis phase. Okay. याच्यामध्ये ग्रोथ आहे होते म्हणून याला काय म्हणतात, नॉर्मल ग्रोथ होते म्हणून याला म्हणतात G1 phase. That's why it is also called as a growth phase. The S phase, S indicates a synthesis of or the duplication or the replication of DNA. That's why this phase is called as a S phase. This takes probably 6 to 8 hours. Okay, 6 to 8 hours. Okay, so that's why this is called as a S phase. This also involves synthesis of histone. Understood? Which are required for the duplication of or the replication of DNA. This is also this part shows the second phase, that is G2 phase. This takes probably, when cell enters in this G2 or second growth phase takes probably 2 to 5 hours. Okay, this is called as a second growth phase or the G2 phase. In this phase, cell synthesis a protein or protein synthesis takes place in this G2 phase. That's why this is called as a second growth phase. In this G2 phase, cell shows high metabolic activity also. So there are several metabolic activity, okay, in this G2 phase. Then this shows also a synthesis of again RNA. Or this G2 phase involves metabolic activities, high metabolic activities, RNA synthesis as well as protein synthesis. This S phase involves replication of DNA as well as histone synthesis. This G1 involves a normal growth of cell and the duplication of cell organelles as well as RNA synthesis. After this G2, cell enters into the actual cell division. That is called as mitotic cell division or meiotic cell division. As I said earlier, okay, as far as mitosis is concerned, mitosis is further divided into two types of mitotic cell divisions on the basis of type of cell, whether the cell is animal cell or plant cell. If it is animal cell, you can see a typical animal cell, then that mitotic cell division is called as a astral type of mitotic cell division, because asters are developed in these cells while division, that is mitotic cell division. That's why this type of cell division is called as a astral type of cell division. What is aster? What do you mean by the aster? So aster, you can see this is called as aster. This is called as aster. A fork-like structure develops in the cell, in the animal cell, is called as a aster. That's why this type of mitotic cell division is called as aster mitotic cell division. This mitotic cell division again, or mitosis, is further divided into two parts: a karyokinesis and the cytokinesis. Karyokinesis and cytokinesis. Karyokinesis, as I said earlier, karyokinesis means division of a nucleus. Karyon means nucleus and kinesis means division of nucleus. Now this karyokinesis is further divided into prophase, metaphase, anaphase and telophase, followed by cytokinesis, that is division of cytoplasm. Now just see what is prophase. So this prophase involves condensation of chromosome, and this condensation reveals into definite number of chromosome. In this phase, each chromosome appears just like a dyad. It means that two chromatids are linked with each other at a centromere. Understood? That's why this also involves a disappearance of a nucleolus and the nuclear membrane. So both these things get disappeared in this particular prophase. Second one is metaphase. So this word itself indicates that is the metaphase. Meta means middle and the phase means stage, in which these chromosomes are organized on a equatorial plane or a center on a spindle fibers. Okay, on these spindle fibers, and these spindle fibers are contractile spindle fibers, proteinaceous spindle fibers, which are connected to the fork-like structures, which are known as asters. Okay, then anaphase. In this anaphase, centromere of each chromosome divides into two chromatids, and these divided chromatids are pulled together towards the opposite poles on the spindle fibers, followed by telophase, that is called as the last or the fourth fourth phase of karyokinesis. This involves late telophase, early telophase. In animal cell, a furrow is, you can see this furrow, and this furrow develops at the center. This leads to the a deep furrow. Okay, this furrow increases towards the center, and at last this cell divides into two daughter cells. In plant cell, this cell plate extends towards the cell wall, results into the formation of two daughter cells. Understood? So this is simply, and the most important thing is nuclear envelope as well as nucleolus, both these things get reappeared. This is followed by cytokinesis, that is formation of furrow, and then the cytoplasm divides into two daughter cells. So this is simply regarding the mitotic cell division takes place in the plant cells and the animal cells. So this mitotic cell division mainly leads to mitotic cell division takes place only in somatic cells, then the body cells, as well as you can say the vegetative cells, while meiotic cell division takes place in generative cells or gametic cells. So this is simply regarding so today you have studied a cell. What is cell? Cell is a basic structural functional unit of life. Then we have studied a cell cycle, okay, including the interphase and the actual division, that is mitotic cell division or meiotic cell division. Interphase is divided into G1, S and the G2 phase, and then the mitotic cell division, actual mitotic cell division, that is karyokinesis followed by cytokinesis. Then we have studied the actual mitotic cell division and the subphases of karyokinesis, that is prophase, metaphase, anaphase and the telophase, followed by cytokinesis. So I hope all of you understood all these points without a doubt. Thank you.